When it comes to fighting off an infection, your body doesn't have time to wait around. It needs a first responder. And that's exactly what we're talking about today, the alternative complement pathway. We're going to break this down step by step to give you a really clear guide that'll help you absolutely nail this for your exams.
All right, so here's our game plan. We'll start with why this thing is such a fast first responder. Then we'll get to know its unique set of protein tools and figure out what actually sounds the alarm to get it started. After that, we'll build the entire weapons system from the ground up, and finally, we'll compare it directly to its cousin, the classical pathway. Let's jump in.
So where does this pathway really fit in your body's huge defense system? Well, think of it like the guards who are always on duty at the gate. It's all about immediate, shoot first, ask questions later kind of action. Okay, if there's one thing you absolutely have to get, it's this. This pathway is antibody independent. That's huge. It means it doesn't have to wait for your adaptive immune system, which can be kind of slow, to build custom antibodies. This is what makes it a cornerstone of our innate immunity. You know, the defenses we're born with, ready to go at a moment's notice. Every system needs the right tools for the job, right? So let's meet the unique set of proteins, the key players, that this pathway uses to get things done. This slide lays it out perfectly. You can see the alternative pathway has its own special crew. We're talking factor B, factor D, and Properdin. And just as important is who's not on the team. It leaves C1, C4, and C2 on the bench. Those are the stars of the classical pathway. Remembering this unique lineup is really, really important. So, if it's not using antibodies to sound the alarm, what does? What actually kicks this whole process into high gear? Let's take a look.
You know, the cool thing is, this pathway gets activated directly by stuff on foreign surfaces. We're talking about things like endotoxin, which is all over bacterial walls, or certain parts of fungi and yeast. It can even recognize our own cells when they look weird because a virus has infected them. Essentially, it's just looking for general danger signals, no specific antibody tags needed.
And that brings us to the main event, the actual mechanism. Honestly, you can think of this as a biological assembly line, where each piece is carefully added to build a weapon designed to just blow up invading microbes. So how does it all begin? With a tiny, random spark. There's this protein called C3 just floating around in your blood, and every so often, it just spontaneously breaks apart. This creates an active fragment called C3b, which is chemically sticky. It desperately wants to bind to something, and if a pathogen happens to be nearby, bam! It latches on, basically painting a target right on the invader. Okay, so once C3b has marked the target, factor B shows up and bonds to it. Then along comes factor D, which acts like a pair of molecular scissors and just snips factor B. A small piece floats away, but the bigger piece, BB, stays attached. What you're left with is this complex, C3b-BB, and that is our first major enzyme, the C3 Convertase. Its job, to find and chop up way more C3. But here's the catch. This new enzyme, C3BB, is super unstable. It would fall apart in just 5 minutes. That's not very useful, right? Well, that's where our third special player, Properdin, comes in to save the day. It acts like a clamp, binding to the whole complex and stabilizing it. This extends its life from 5 minutes to a much more useful 30 minutes. It's an absolutely vital step. Now that it's stable, that C3 convertase goes into total beast mode. It starts rapidly chopping up hundreds, even thousands of other C3 molecules. This creates a massive amplification loop, coating the pathogen in a ton of new C3b fragments. And then something really cool happens. When one of these new C3b fragments binds to the enzyme complex itself, it gets an upgrade. It becomes C3b-BB3b, and that is our C5 convertase. So from here on out, all the complement pathways merge and follow the same road. The C5 convertase does its thing. It cleaves a protein called C5. The C5b piece then kicks off the final assembly line, calling in all the troops, C6, C7, C8, and a whole bunch of C-nods. Together, they build the ultimate weapon, the membrane attack complex, or MAC. And this is the end game. The MAC is basically a molecular drill. It punches a hole, a big pore, right through the pathogen's membrane. Water floods in, the cell swells up, and pop, it explodes. We call that cell lysis. It's a direct, brutal, and incredibly effective way to kill an invader.
All right, to really hammer this home and make sure it sticks for your exams, let's put the alternative and classical pathways right next to each other for a quick, high-yield comparison.
This table just lays it all out so clearly. You can see the big differences right away. Innate versus acquired immunity, what kicks them off, and of course, the totally unique enzyme names. Seriously, Being able to identify C3BB as the C3 convertase for the alternative pathway is a classic exam question, so lock that one in.
Okay, so let's boil it all down. What are the key takeaways? Number 1, it's antibody-independent innate immunity. Number 2, its key players are factor B, D, and Properdin. 3, it's kicked off by foreign surfaces. 4, its C3 convertase is C3BB. And finally, its whole purpose is to create that membrane attack complex and make the bad guy go boom.
If you like this video, then please like, comment, and subscribe.